You have this either kind of an either or situation, so I paired them up based on how do microbes get their carbon, what do they do? Is it carbon dioxide and then they make their own sugars, so they're autotrophs? Or are they like us, where they have to take something around them that was once living and take that carbon source and use it? For energy, we have to use chemicals, so we're chemotrophs, but plants and cyanobacteria and some other microbes could do light energy instead. And then this idea of where are they getting their electrons? Is it some organic molecules like what we do, or is it being stripped off of some inorganic? So learning these in pairs and understanding that this is like a huge concept. All microbes, all living organisms, including us, we have to get carbon, energy, and electron sources from somewhere. And so these are the options. And then, as I said last time, you can start taking these terms and combining them and get interesting groups of microbes that we know what they're doing here. So if we say, oh, that's a photolitho autotroph, you have to pick apart the word and look at it and say, okay, photo means light, litho is rock, so that must mean inorganic electron source. And then autotrophs, they're making their own sugar, so CO2 must be what they're bringing in. And so we do have quite a bit of microbes that fit into that category. And then we looked at this one, the chemoorganoheterotrophs, which we fit into this. That's what we are. And most bacteria that have been isolated and actually studied carefully, they fall into these two categories. But then there are other groups of microbes that are falling into, at least so far, there are not as many of these. So photoorganoheterotrophs, they're a little weird because they'll use light energy, but they also use organic compounds as an electron source. And then they often use an organic compound for their carbon. So they're not bringing in CO2. And at least a lot of them, some can, but a lot don't. So they're a little weird. And you often find them in polluted areas because that's where a lot of organic compounds are located and light. So polluted lakes and streams are excellent area to go if you're trying to hunt for photoorganoheterotrophs. And then chemolithoautotrophs are very, very important because that's the group of microbes that are so heavily involved in recycling our nutrients on Earth. And so here, chemical as their energy source, inorganics as their electron source, and then they're taking in CO2 for their carbon source. And so a lot of microbes are doing that. And we have them to thank for basically how the Earth formed and all the chemicals and minerals and recycling going on. And then this table, I was talking to you last time, how sometimes I've had some students say they like it this way, where they can think about it in this, it works better for their brain. So whatever way you want to look at it. But this is basically saying what we just talked about in the previous couple slides. Okay, so I think, I hope I've convinced you that microbes are incredibly diverse in their physiology. And like I said, when I was a grad student, I had to take a whole year of physiology of microbes. So we spent what, 2 weeks on it, 2 weeks. So obviously I'm leaving out some things, but I'm trying to give you some big picture highlights of all the diverse things that they're capable of and how amazing they are in terms of their physiology. They're super well adapted to being able to adapt to their environment, even as their environment is changing. They're very versatile, much more so than we are. We don't change and mutate like they can. Questions on the physiology stuff? We went through a lot. But remember, don't get bogged down in all the details. Don't get lost in that. That's not what this section is about. We good? Okay. I'm going to shift over. Okay, so on your syllabus, theoretically, we're supposed to be getting into microbial habitats next, but I want to save that. I'll push that a little later. I'm going to get into methods of how to sample. What kind of methods do you use to sample an environment? I'm doing it now because you need this information for your research project. So as you're like thinking about your research and kind of thinking about, well, what do we want to do? And then how are we going to tackle that? What are we actually going to do? Like, how do we take a sample? How do we work with it? What do we do once we get it back to the lab? So that kind of information would be helpful now rather than later. So I'm going to switch over to that. So that should be available in EDUCAT, I believe. And so this is a big topic. It is something you have to consider carefully before you actually run a big experiment, especially if it's a massive experiment that costs a lot of money. You got to think through this very carefully first before you actually go and do it. And, you know, there's all kinds of habitats that you may want to go sample microbes from. And depending on what kind of sampling you're thinking about, the methods can vary tremendously. Lots of beautiful areas. I wouldn't want to physically sample this, but this is one of the hot springs out west in all those beautiful colors are primarily cyanobacteria and some algae. That's an aerial photo. I actually had a student, a microbiology major who did a summer internship out in Montana, and they got to go and sample the hot springs. And they super careful and get all kinds of permission in order to do that, because of course, this is very dangerous. So she didn't do this. This was a picture in a very famous microbiology textbook. And I just laugh every time I look at this, that they put it into a textbook. So my interpretation of this is the master's student. This is the postdoc or PhD student, and that's the professor. Standing there well away from the risk. Here, this guy's holding on to this guy. So he won't fall in as he's taking a sample. And it looks like it's duct taped. I don't know, maybe not, but I like to think it's duct taped. So this is not what I'm encouraging anyone to do. I just giggle every time I see that. So taking samples from hostile environments can be very tricky, and the technology has really lagged for a long time. This is what you were doing, and we don't allow that anymore. But you know, anytime you approach a habitat and you're trying to look at it from the microbial ecology perspective, some of the questions that you might be asking is, you know, what do we do to find out what kind of microbes are there? And then, you know, what are they doing? And how do we figure out which microbes might be really impacting that system that you're looking at? And so these are very commonly asked when you start approaching this type of research. And then we have to appreciate that when we start working with any samples in the lab, we have to keep in mind that what they're doing in the lab may not be what they're really doing in nature. Remember, they're super versatile, and a lot of microbes have a lot of genes available to themselves where they can do something different in the lab that they really wouldn't do in nature, in their natural habitat. So we have to think about that. The other thing we really have to kind of warp our mind to think about is that we have to think really, really small. So scale issues, humans versus microbes. This is a good analogy. A million-fold difference in size between us and the standard microbe. So a really good analogy I read that I think is helpful. A 3 micrometer or micrometer rod shaped bacterium living in a habitat that, let's just say we go out 3 millimeters from that cell. And then we do that, you know, what does that equate to? It's equal to a human and a 2-kilometer distance around them. Weird. That's kind of mind-bending. So 3 micrometers, 3 millimeters is equal to 2 kilometers for us. So this is trying to get you to appreciate that these bacteria, the microbes in general, a little bit of a shift. You know, we think 3 millimeters is tiny, but to them, it's 2 kilometers in the, you know, equal analogy. So here's an example. Visually, this is a soil particle. You're looking at a soil particle. And here you're looking at the distance, if they go for the center, they call that, you know, the 0 point. And then as you work your way out, you're going certain millimeters away from the center. That's what they're showing on the x-axis and on the y-axis. And what this different color coding is availability of oxygen for any microbes that might be lurking in or on that soil particle. So you can see that the percentage of oxygen can vary quite a bit in just a millimeter or 2 distance. So I think that's kind of a nice analogy to think about. I used to think about this. I used to get rid of when I took Gen Micro as an undergrad, I'm like, my professor said that you have a lot of anaerobes in your mouth. And I'm like, wait, what? Anaerobes? They don't like oxygen, but I'm breathing oxygen all the time. How can that be? And then once he explained this, like, oh, so they're in the nooks and crannies in your gum area. And for them, they can easily find a spot in your mouth that has no oxygen. It's not a problem. But again, that scale issue is really hard for me to think about that initially. So give that some good thought. So very often what we do when we take a sample and bring it back to the lab is some kind of enrichment technique where we're perhaps looking for a certain group of microbes. And so we need to process that sample to enrich for that group of microbes we're looking for. So this is what it's trying to show here in this mock-up is the idea that you get your sample, okay, looks like a soil sample, and they have some kind of broth in the flask with some recipe. So certain types of carbon, energy, and electron sources in there. They dump their sample in, and then they probably incubate that for a while. And what they're showing is a theoretical species mixture in that sample. So they say, you know, let's say that's what we started with on the top bar graph. And you can see, you know, it looks like species D wins, more abundant in that sense. E is not very abundant. And then they run their sampling through this process. And now you can see the bar graph below. If you compare that to the bar graph above, big difference. So who did they enrich for? Which species? E, you bet. They're enriched for E. Maybe knowingly, maybe unknowingly. You know, so sometimes things can happen that you really didn't plan for. That's not what you were going for. So all of this requires deep thought before you go and do it, because you really have to think about your question. What are you trying to answer? And then what's the best strategy to get some data that actually represents what's going on in that habitat? Not what they can necessarily do in the lab, but what can they do in that habitat? So then this brings up this idea of the so-called Heisenberg uncertainty principle, which I think most of you are, you know, at least somewhat familiar with that phrase. And what this is getting at from the microbial perspective is basically when the closer you examine microbes, the more likely you're going to introduce some kind of artifact into that system which could bias the information. So you just have to be aware of that. And when you do microbial research, you know, we're all aware of that and consider that as just a reality. So anytime you go and, you know, take a sample, you are definitely disturbing that habitat. And remember, a 3 millimeter distance for a microbe is huge. So you can do some massive disturbance. And to you, it doesn't look like much, but to them, it's a lot.
Can you refer to artifacts, for instance, contamination, essentially, or like what do you do?
That's part of it, but that could be one way of what introducing artifacts, or it could be even just something like you go to take a soil sample, and as you're doing that, you accidentally mix that soil sample just by taking the soil sample, and now you've changed their world,
Right.
really, so. That would be an artifact too, because now you've really changed how they were living because of your physical disturbance of, maybe it's a soil system. And then, of course, anything you do to manipulate those samples might change their behavior. Remember, microbes are very good at adapting on the fly. They can turn genes on and off on the fly. They're constantly assessing their environment, and they're trying to figure out how to survive any shift. And hopefully they have the necessary genes as kind of a reservoir inside them that they can, okay, and now I need to turn this gene on, I can turn that one off, and because this situation in my life has changed. And so you have that, and anytime you take a sample, you've now changed the habitat. And again, keep the scale in mind. It can, to you, it may look like no big deal, but to the microbes living there, it could be pretty massive. Now, when I first started doing microbial research, especially when I was planning a huge experiment that was costly, especially doing for grad research or postdoc research, it can almost get paralyzing thinking about this, because you're like, geez, no matter what I do, you're going to be messing things up, so to speak. But again, we all recognize that you can still publish great data, get great data, good things to think about, make progress in new science, but you just have to keep this stuff in mind and be aware that there are limitations. And you don't exactly know, here they're looking at like, as soon as you take a sample, so time zero is as soon as you take a sample, do things immediately start changing? Or when you take a sample, do things cook along for a while pretty much the way they were, and then something happens and you start getting a shift in things and artifacts? And so you don't know this, you don't know. And so there are things that you have to be aware of and know that there are limitations to how you interpret your data, I guess would be the way to say it. So what kind of sampling are you going to do? When you go to that habitat, are you concerned about what's in the air? Is that what you're going for? Or maybe the water or the sediment of the stream or lake? Are you going for soil samples? Here they're just saying some very basic ideas of, if you are taking air sampling, and by the way, they call that field of microbiology aeromicrobiology, and it's becoming more and more, that used to be a very tiny field of microbiology, like not many people were studying that, but now, especially with the pandemic, there's more and more that research going on in that, in terms of how many microbes are in the air or spores are in the air, how are they traveling, and they're traveling globally. When microbes are spewed up into the air system, maybe from earthquake or something like that, wildfire, you do get global transfer, and they can show that. But anyways, you're seeing that, usually your numbers are pretty low and not highly concentrated. And so if you're taking an air sample, you would grab your air sample, but then you usually have to concentrate your air sample. And so you usually suck it through a filter, like what you did on Monday's lab. Instead of water, you would have an air sample sucked through a filter. And then you would concentrate your microbes. Hopefully they'll grow on an autoclave, for example. And so Anderson samplers, we were working with those a little bit when I was a postdoc. It's just a fancy piece. It's like a fancy vacuum, essentially. Takes a suction of air and captures it. And then water, it can be, depending on what water source, you might have a lot of microbes or not very many. And so you might have to dilute your sample, or you might have to concentrate it. Another way to concentrate, like water, if you want to concentrate a water sample, often that involves centrifugation, where you take your sample and centrifuge to get a pellet and then resuspend in a smaller volume of liquid. Sediments and soil usually have high numbers, so you usually have to dilute. And so you would use the common strategy as a spread plate dilution where you severely dilute your samples. You get your soil or sediment into water, dilute it, and then plate it out. So you've already done that. So this is two examples of water sampling. You can use Van Dorn bottles, which are often used for like algae or cyanobacteria, protozoa. So you can get that to come down to a certain depth in the water, and then you release what's called a messenger, and it will pop open the two openings there. The water goes in, and then you can pop it shut. So you take a sample at whatever depth you want. And this is a similar kind of old-fashioned technique, the JZ sampler. So basically, though, they have ways to, like, I want to sample at 1,000 feet. So you don't put like an open bucket all the way down to 1,000 feet, because then you're getting the water from 0 to 1,000. You want water at 1,000 feet. So you want a closed container, get it down to 1,000 feet, and then open it, and then shut it and bring it up. So they have these types of things for sampling, for water samples, especially out in the ocean. Okay, so this is a flow chart. I'm gonna break it down. So you go to your field site. Let's say you are, I'm gonna say we're gonna sample some soil. And that's what this image is. So these are cork borers. You might have worked with those in various labs. And you may have noticed that there's different sizes of diameters of cork borers. And so when we were when I was a postdoc and we were doing experiments on looking at microbe interactions, we were very interested in, do our Streptomyces bacteria, how closely are they hanging out with each other? And are they able to communicate with each other? And so we wanted to capture soil samples that were meaningful at their scale. Remember, 3 millimeters out is 2 kilometers. So the ordinary cork borers that they sold were too big for what question we were asking. So we actually had the university manufacture really small cork borers so that we could take core samples and we could say with confidence, this core sample should represent microbes that are hanging out with each other and interacting, because it's a scale issue, a tiny diameter. And so, we had to really think about that before we started sampling, because one of our big questions was which microbes are interacting with each other, which aren't. And so we were trying to make sure that we got neighboring microbes to see how they were interacting with each other. And then, also asking how much should you take? And then especially if you do repeated sampling at the same site, if you take too much, you're really disturbing the system. Take too little, maybe that's not adequate for your answering your question. So you have to think about that. So the capture idea, there's lots of different ways to capture your sample. And you might not care that they're alive, so you could immediately fix them into a stable state, but that usually kills them. Or you could say, well, I want these microbes alive so I can study them back at the lab. But then you can't just like stuff them in your car and go home. You need to put them on ice, most likely, or well, yeah, usually we just bring like coolers. We have coolers with ice packs, and we would put our samples in there. Well, okay, so then if you are planning to fix your sample, which means usually kill it, maybe you're just going for their genes, you're just going to do DNA sequencing, you don't care that they're alive or dead, then you have to do it quickly. Storing is a big deal. How do you store these samples before you get back and can start working with them? And so usually you keep them cool or frozen. Basically, what you're trying to do is stop any changes from occurring, especially their gene expression patterns. You are trying to keep them quiet and like, just stay the way you were, as you were in your habitat. I want to keep you that way. That's very often what we want. And so time is definitely of the essence. So when you take your sample, you ideally, in an ideal world, you go back to the lab and immediately start working on that experiment. But that's often not the case, so you do it as soon as you can. The longer you wait, you can't be as confident in your data because changes happen. So many changes can happen. Even what kind of container you choose. Like I'm gonna go in a few minutes to get you some plastic bottles. Well, plastic bottles versus glass bottles, which would be better. I mean, they've even looked at that. Glass bottles, they've done some studies with that. And so the water samples that we get, often they adhere to the wall of the glass. They like to attach. Microbes are kind of fond of attaching to surfaces. And so over time, the water the microbes in the water will start to adhere to the inside of the glass. And so that's why we always tell you, swirl the bottle before you take a sample. And then don't forget, they can reproduce phenomenally fast. E. coli, if you give E. coli everything it loves, 20 minutes is all it needs to double its population. So they can replicate very rapid, which causes a shift. And then some microbes are better at others. So if you have a mixed population, some, just like in that enrichment bar graph, some microbes can just like, yeah, I'm going to reproduce like crazy under these conditions. And others are like, yeah, I'm going to slow down. And so you start to get these shiftings of the population. And then when you actually go to analyze your samples, maybe you're going to do some microscopic work. Maybe you're going to extract some things and study them biochemically or physiologically. But basically, you want to think about how are you going to process those samples based on what you're going to do with them. You have to give that a lot of thought. And then you have to be careful that you're constantly mixing so that, they're not adhering to the bottle that you have them in. Or with soil samples, very often what we do is pop the soil into some sterile solution. So that's the diluent. And so we're thinking, you even have to think about that. Like, should you put them in sterile water? Should you put them in phosphate buffer, some other kind of buffer? How should you do that? And then what you want to do typically when we take soil samples, we want to say, like very often when I was doing experiments, I would ask how many Streptomyces bacteria are in the soil sample. And so when you are mixing the soil with whatever solution you put them into, you have to think about, well, how long should I do that for? How vigorous should I do that for? What temperature should I do that for? Do that at? And so you have to think about all of this if, based on what the question is you're trying to answer. I was looking at, I want to know how many Streptomyces are in that soil sample. And I was going for which ones are actively producing antibiotics. That was one of the things I was asking. So thinking about all of this, generally you think about, okay, well, where does the microbe live? What's the temperature of that habitat? That might influence then what temperature you pick. You're trying to keep things pretty similar to their habitats. Revolutions permitted of mixing is getting at the idea of if you mix that soil sample, what you're doing is helping to agitate the microbes off the soil, which is what you want. You want this microbes hanging in the solution and not attached to the soil. You're also introducing oxygen into that solution as you mix. That's one of the reasons we do that. So there's a lot to think about. It's a little bit overwhelming. And so anytime I set up an experiment, I have to think about this more than just a day in order to be confident that I have a good strategy. It takes a lot of thought. So when you do microbial ecology, you really have to there's a lot of forethought that goes into this before you actually do it. Okay, so here is just this idea of mixing time. They took their soil, put it into some sterile solution, and they put it in a blender and was mixing it. 2 different soil samples from 2 different areas. And then they did a serial dilution like what you did. They used their glass spreader, spread it out, and then they incubated and looked for colonies and counted those and got a population estimate. So I'm gonna let you think about that. I'm gonna go grab those water bottles. I'll be back in just a minute. Hmm. So quickly to finish this thought, you can see they've asterisked the strategy that got you the most microbes. So they were asking, they wanted to know how many microbes are in the sample. And so they were trying to find a method that would try to get as many microbes as possible isolated and grown up. And you can see it, a 2-minute blending for soil 1 worked the best, but an 8-minute for soil 2 worked the best. So that's not terribly encouraging. Means there's no like one way to do it to really any soil sample. Yes, it's gonna work. No. And so there's these types of issues that you have to think about. But let me just pause for a second. Forgot to pause. Pause. All right. One thing, so we have three water samples that I need volunteers for. So for this physiological profiling, be nice to get water samples that we know for sure there's gonna be microbes there, but slightly different in terms of the water quality or water habitat that they're taken from. Anyone want to volunteer? Duck pond, that would be a good one. We did that before and it worked out well. Duck pond. Chocolate Bayou would be good. No, it's not. One more. Okay. Yeah, bigger lake. Thank you. All right, so just make sure that you tell Duncan where you sampled, and then we're gonna do the um physiological profiling for that. So let me just here, I'm gonna go back to. Oh, never mind, I'm still recording. I didn't pause. I thought it paused. Okay. Here, let me just finish this thought. Here, they're looking at, okay, if you take a soil sample and you're trying to get as many bacteria out of that as possible, what should you pop the soil sample into? Should it be tap water, pyrophosphate buffer, Ringer solution buffer, Wieland-Grasky solution buffer? And again, you can see a big difference in terms of what they ended up with. Viable count means they put them, they did the spread plate technique, like what you learned with the glass spreaders, spread it around, incubated, and whatever. Viable just means then they grow, they're viable, and you count those. And so looks like Wieland-Grasky won, right? That buffer, you get the most. So again, even something like that, what kind of buffer should you pick can make a big difference. So it's enough to, kind of make your head hurt when you're starting to plan this, but it's important to think through it. And I think, again, if you keep in mind the scale idea of, what a habitat is to a microbe in terms of that 3 millimeter difference can be like a 2 kilometer difference to us. And then start working through this, just methodically work through how to approach sampling. All right, I think we'll stop there and we'll continue on with that idea tomorrow and get more into this whole process.